Hey Rebels, my name is Matthew Barton. Welcome to the Rebellion Brewing Podcast. Today is a momentous day for the show. We're recording episode number 200. It's only fitting that a podcast about beer gets into the weeds of something so essential to brewing beer, we don't even fully understand it yet, and that's yeast. Professor Chris Eskiw from the University of Saskatchewan joins us today to talk all about his research and its impact on the future of beer. So let's get into it. Chris, welcome to the show.
Thank you very much for inviting me on. I was super excited to get the invitation and happy to share a discussion about what's going on in the lab.
How's it going in February for you?
Not too bad. Not too bad. We've got some exciting news the last couple of weeks. We found out that the province is supporting our research into investigating the links between yeast genes, yeast genetics, and the brewing process. So super excited to have the province on board and have so many people interested in what we're doing in the lab.
To give people a sense of what that research means, what is your background in this kind of work? Like, what do you do?
Well, this will be interesting because I'm traditionally, my research program isn't focused on beer initially. So after completing my master's degree at the University of Saskatchewan, I went on to the University of Toronto to get a PhD and then moved on to Oxford University and then Cambridge University, studying the relationship between our genetic complement called our genome and how that interacts with our environment and how that interaction influences health and longevity in humans. Now, how this becomes extremely relevant is that interaction between your genes and the environment. There is communication there. And even if your genes aren't behaving properly, you can modify that behavior using the environment, like nutrients. And this is exactly what goes on, in my opinion, with the yeast during the brewing process. Even though you may have a certain set of genes per yeast strain, you can modify the behavior of that yeast by changing the environment in which they live and express the right genes at the right time. So this is where I'm coming from, and this is where I'm going to. And it seems like a crazy link, but it fits really well together.
So you started off studying how people age, or how the environment impacts people's aging?
Absolutely. So my major research here at the university is looking at human longevity, health span, and lifespan, and how diet actually influences that through modulating your genes. So I'm very much interested in genomes and genes and how they interact with the environment.
So how do you go from genomes and genes and aging to brewer's yeast?
Excellent question. So I had a talented student who finished up a master's degree looking at basically how the genome folds inside cells. So it's basically genetic origami, how your genes are folding together and how that influences how your cells live. And after he completed his degree, Josh Pickering, he moved on and wanted a little bit of a change from that research. So he stumbled onto Casey Murray and got a job at 21st Street Brewery here in Saskatchewan. And after a couple of months, Josh and I got together for, of course, a beer, and we started talking some questions, some research questions. And what fell out of that conversation was, what are the yeast actually doing? How are their genes actually contributing to this process of brewing and creating all these wonderful flavors that we're getting in all the different styles of beer? So that's pretty much how this came about.
Well, that's pretty heady stuff. A little beer pun there for you. What questions are you asking with this work?
Okay, so the main question that we wanted to address was the idea that, again, the yeast are all different. There's different strains. You have your ale strains, you have your Belgian, you have your lager strains. And essentially they're taking those starting components that are in your wort and converting them into not only your ethanol, but all those flavor compounds. And the question was, okay, for making an ale, you use that ale strain, but what makes that different from a lager strain? I know there's temperature, there's nutrients, all those things are different, but really what drives them in the background. Now, that collection of genetic material known as the genome is the program that tells these yeast how to behave. And so out of the couple of beers that we had, that question was, okay, how are these different? Why are they doing these different jobs? And the next obvious question for us was, okay, which genes are actually present in those strains that make them unique? How are they selected over evolution? And how can we actually utilize this information to brew better beers more consistently and generate more diversity of beers?
We know that when we're brewing with our big batch system, we can only reuse or repitch yeast a number of times before it's mutated way beyond what we can do with it, and then it starts creating like off flavors and producing stuff in the beer that just makes it undrinkable or terrible. And we have to start fresh with what they call clean yeast, or yeast that's been produced in a lab in very specific conditions. So what I heard from you there was you might be able to maybe talk about a stabilization of yeast or something?
Absolutely. So that process that you're talking about, only being able to pitch yeast so many times, this is actually the equivalent of human aging. So those pitches, the cells get older and older, and those cells forget what they're supposed to be doing. So there is, again, another parallel between what's going on in humans and what's going on in those yeast. So as they get older, they forget what they're supposed to do, and they don't grow as well. What we're aiming to do in the lab is actually rejuvenate those populations and develop tools or methods in which brewers don't have to go through these steps where their yeast goes off and they end up with an inferior product. How can we actually get you guys to have your brews that are more consistent and yeast is no longer a problem?
Consistency, man, that's a game changer. That's huge.
Yeah.
I know our brewing team spends so much time thinking about yeast, managing yeast. We spend tens of thousands of dollars every year, and it's a significant cost and thought piece. To have something more reliable would save us so much money. It would help us bring down costs. It would, wow, that's kind of neat.
And even for the home brewer, if you have your collection of yeast at the end, how can you best store that and then bring it back up to speed before the next time you repitch it? So even your home brews will be more consistent. That is what we're aiming for.
Well, I know that you were working with 21st Street, as you said. So what have you learned from those guys?
Well, like I said, when I first came into this, I was pretty much a novice. So spending some time in the brewery with Casey and now my PhD student, Fina Nelson, and going through the basics of that. But what we've learned is that when you propagate the yeast, when you start getting their numbers back up, using different nutrient conditions will actually change how that yeast performs when you pitch them. We did a pilot experiment taking some, I do believe the strain was 1318, and just growing it up under 3 different conditions to get our pitch rate equivalent. We put those yeast into the same wort, and we ended up with 3 different beers with 3 different colors and 3 different flavors. Genetically identical yeast, but because they were propagated a little bit differently, they were behaving differently, and it gave us different results, which was super cool. We were super excited by that.
How did the beer taste?
I think all 3 were great. I prefer things that are a little more hoppy and a little more full-bodied, a lot of flavor to them. where some of the other preferences were for the non-hoppy, a little more towards the IPA side. But all three were very acceptable. We did some preliminary sensory evaluation tasting, unofficially in the hallway, to anybody who wanted to stop by and have a taste. And yeah, the results were great. And it was striking how much variation we got from just that little bit of growth.
So if you've been doing these kind of tests and these trials, very early on in this interview, you said you've got approvals for a new project or some additional research. Tell me about that.
So the funding that we've received is just an extension of that. So those were a couple of pilot experiments, but now we want to investigate what really made those three beers differently. Now, the strain was the exact same thing, so it had the exact same sets of genes. But what's important is not only having genes, but knowing when they turn on and when they turn off. Timing is extremely important. And your cells can turn genes on or off like dimmer switches on light switches. They can be on a little bit, they can be on a lot, they can be on at the right time or the wrong time. So having a gene is important, yes, but knowing when that gene is on or when it's off is also important. So our new investigation is to map out when these genes get turned on and when they get turned off, and how that relates to the flavor profile of those beers. So when gene A gets turned on, does flavor compound X also appear? And now we can say that this gene produces this flavor. We can control the timing of that and give you the flavor you want when you want at the appropriate levels.
I'm going to lay out the way I'm thinking that you said this, and you can tell me if I'm wrong or you can clarify.
Sure
right
thing.
Now, as it stands with hops, we know that we've been talking with hop scientists, and they say if you put hops in at this temperature, you're going to get this. If you put hops in it at a lower temperature, you're going to get that. If you use a hop back or you use a hop torpedo, you're going to get these different expressions of flavor and reactions, or you might get more bitterness. And so understanding that happens from a scientific perspective gives brewers extra tools and more weapons in their arsenal to make a really kick-ass beer. What you just said to me sounds like we can do with yeast what the hop scientists were telling us 15, 20 years ago that we can do with hops.
Yeah. And with the hops thing, that's the extraction of that material. But for the yeast, we can set up conditions where the yeast are now tunable, like you said. And you can say, I have strain 1318. I'm going to treat it this way, and it will give me this flavor profile. Or I'll treat it this way, and it'll give me that flavor profile without having to genetically modify the yeast or buy a new strain.
That's really cool, man.
Yeah. That's the goal. And from what we've seen so far, this is what we definitely, we feel very strongly about that. But of course, now we have to test that. And we're doing lots of work to make those associations. And once we're done, our plan is to create a handbook to say that you have this strain, grow it this way, you will get this flavor predictably. To try to take all that guesswork out of what's going to happen.
Really? And will this just be available to the public, or you're going to publish like a big tome that kind of like brewers will only have? It'll be like a hidden secrets, or how's that going to work?
We actually, our hope is to make it publicly available. We don't only want to help businesses, but we want to help the home brewer to make those connections and make this information freely available. Now, of course, your recipe for your beer will strongly, what goes in will strongly influence how those yeasts behave. But because of the yeast, their cells, their biology, they're a little bit sloppy in what they do. If you can control that sloppiness or that variation in what they're doing, that will give you more chance of success with what's going on. So that's our hope anyway.
previously on the podcast, we've had episodes dedicated to Belgian yeast and Kveik yeast. We know that there's a specific yeast called Cerveza, which is how the name Cerveza comes to be. You find it in Spanish for beer. Could there be a Saskatchewan yeast that comes out of this project?
Absolutely. One of the things we're thinking about is tapping into the vast resources of yeast that we have in our natural environment. For example, Hascat berries, which are popular fruit here in Saskatchewan, they have a wild population of yeast growing on them. And you can take samples of that and try to use it for brewing. Now, it's a little bit hit and miss because wild yeast don't like to do what our domesticated yeast do. It takes a little bit of convincing. But we're also planning on devising tools to screen the yeast and say, okay, you isolated this should make a great beer. This one should give you some funkiness that maybe you will not enjoy so much, so maybe hold off on that one. And use it to identify these new strains that might be laying around in the Saskatchewan environment and use those to create novel Saskatchewan products.
That's deadly. That sounds like you've connected our farmers to the brewers, science to the brewers. You've created a whole new industry where we're going to need people who are smart at lab work, people who have that background and understand all the science and the research that you're putting in. That sounds like so many opportunities to build up this industry, make it something special and unique, build our economy. That's really exciting.
And that is definitely what interests me. But I also know I've given some complicated explanations potentially to what a genome is. I want to take the requirement to have that kind of knowledge out of it and say that, again, when you look at this particular strain of yeast, it has the ability to uptake, let's say, extra maltose or maltotriose. You're going to want to use it to generate this flavor. And we know that because it has extra copies of those genes. As a brewer, you don't need to know that. You just want something that will absorb or utilize that maltose better. So to create that handbook and say, hey, this is what you want to use to generate this. Here's how you grow the yeast, and it'll give you a very consistent product. Take all that guesswork out of it for you without having to become a PhD molecular geneticist. So
it sounds like you've taken the approach of computer scientists where they said you don't need to know coding or binary. You don't need to know HTML, but you can surf the web. You don't have to figure out how GPS works, but your cell phone will work in your hand and like a child can pick it up and boop.
Exactly. Yeah, we want to make it that simple.
That's so deadly. Man, if people have been listening to this podcast and they want to learn more, they want to figure out what your team's up to, or they just want to ask you questions, where can they go?
Well, you can check out my website, www.eskwlab.com. That will give you some starter information on what we're doing. You can also find us on Twitter. Chris.Eskiw is my Twitter handle. And Facebook, you'll find me on the Saskatchewan Beer Lovers page, the Canada Beer Lovers page. I've been getting involved with all of these really cool beer social media pages and learning a lot that way. I think that's probably the best way to do it. There is a lot of really fantastic scientific information, but again, it's a bit on the heavy side. And I wouldn't recommend some of those publications. I'm struggling with a lot of those, so it's really cool stuff.
You previously mentioned your students are really plugged into craft beer and brewing. Tell me a little bit more about that.
All right. So I've recently picked up my first PhD student to work on this project, Fina Nelson. And Fina is really interested in promoting women in the brewing industry. She's noticed that there isn't a lot of ladies doing this. And so what she is planning on doing is organizing a ladies' brew night to celebrate International Women's Day. And this is going to be on March 8th at 21st Street Brewery. And she'll be guiding ladies through a full-scale brew and how to set this up. And this will be done in collaboration with Leanne Katischuk as well, who's a certified beer judge. And they'll also be hosting a beer tasting night to, again, try to promote not only the brewing industry and brewing science, but women in brewing.
I'm super glad that you got this new research project going. I can't wait to bring you back in a year or two to find out what happened and learn more. Thank you for your time today, man.
No problem. I would like to finish off by thanking some collaborators. So the folks at 21st Street and the Senator Pub here in Saskatoon have been fantastic. We've also have on this collaboration Maker's Crafted Malt has contributed to this as well. Shepherd Farms we've been talking to them about their hops and what they're doing in the background, even though it's beyond the scope of this current research. Certainly want to get involved with those folks in the future. And once we are a little more comfortable with our findings, we definitely want to meet up with the rest of the brewers in Saskatchewan and talk about how we can best disseminate our findings and make this available to everybody and make some wicked beers. Really looking forward to doing this.
Make some wicked beers. That's the best way to end the podcast. Rebels, thanks for listening today. If you have any questions or comments about this episode, be sure to join us on our brand new Facebook group page, The Rebellion Brewing Podcast. Those links that Chris mentioned, I'm going to include them in the show notes. Be sure to check them out. I'm also proud to let you know we're members of the Saskatchewan Podcast Network. It's a one-stop shop for tons of locally produced shows from across our province. You can find them at saskpodcastnetwork.com. Be sure to follow us on Facebook, Instagram, and Untappd so you don't miss out on the latest in Sask craft beer news. Thank you for joining the Rebellion.